Good evening, students. So welcome you all to another session, right? So in the previous session, we started discussing about the topic called as the mechanism of evolution. So we said that we'll be discussing two important mechanisms pertaining to the process of evolution. One was deviation from Hardy-Weinberg principle, and the second one is the evolutionary forces, right? So with regards to the first one, we have already studied about it, right? So we took up with the Hardy-Weinberg equilibrium. We saw what the Hardy-Weinberg principle states, and along with that, we also saw the Hardy-Weinberg equation, right? And we have solved a couple of problems also pertaining to the Hardy-Weinberg equilibrium, right? And in the previous session itself, we started off with the second mechanism of evolution, right? The second mechanism of evolution, which was called as the evolutionary forces. So we began with this evolutionary forces pertaining to the mechanism of evolution, right? So before we start off with today's session, right? So before we start off with today's session, let us have a quick recap of what we have seen in the previous session, right? So we said that what do we mean by evolutionary forces, right? So any sort of a force or factor which brings about some sort of changes in the gene pool of a population, we started calling it as evolutionary force, right? So what is an evolutionary force? Any force or factor which brings about some sort of changes in the gene pool of a population is called as an evolutionary force. So I just gave you the list of the evolutionary forces, also what we'll be discussing in this particular session, right? So pertaining to that, the first evolutionary force what we have is the natural selection. The second one is, we call it as gene flow, or we also call it as gene migration. The third one is genetic load, what we'll be talking about, and the fourth one is called as genetic drift, and the fifth one is mutation, and number 6 would be with regards to the topic called as genetic recombination. So these are the 6 different forces, what we call them as evolutionary forces, which bring about the process of or which would bring about some sort of changes in the gene pool of a population, right? So we in the previous session, we began with the first evolutionary force, which is called as natural selection. So we said that there are 3 types of natural selections what we'll be studying. The first type of natural selection was called as the stabilizing selection, what we have already studied, right? So what is the stabilizing selection all about? When does the stabilizing selection operate? I told you this, right? So the stabilizing selection usually operates in a stable environmental condition. The foremost thing what you need to remember pertaining to the stabilizing selection is where it operates. In which sort of environment does this sort of selection operates? So this sort of a selection, which is called the stabilizing selection, usually operates in a stable environmental condition. So what happens in this particular process of stable selection? So we saw that all the organisms, or rather the individuals, attaining the mean phenotypic value, right? So we also put it this way: all the organisms which are said to be present in the peak region of the phenotypic distribution curve, or we can take it this way also: all the organisms which are said to be fit, they alone are being selected by the nature. Whereas the organisms present, or the individuals present towards the either sides, or rather either both the extremes of the phenotypic distribution, are eliminated from the population. So this type of selection, we started calling it as the stabilizing selection, right? So we also made one more point also with regards to stabilizing selection, right? So I also told you that in this sort of a selection, there would be no evolutionary changes occurring in a population. Why there is no evolutionary changes? Because all the organisms which are said to be fit, all the organisms possessing the mean phenotypic value, they alone are being preserved or being selected. So there is no evolutionary changes happening in this sort of population. But this sort of a selection would always maintain a stable phenotypic characters throughout it, or rather from generation to generation. That is the significance of this sort of a selection. So that was the first selection what we saw, which was called as stabilizing selection, or we also called it as the centripetal selection, right? So in this session, let us take up with the remaining 2 more types of selection under natural selection, and we'll get into different types of evolutionary forces what we have listed out in the previous session, right? So the second type of natural selection what we'll be talking about, right? Second type of natural selection, what we'll be talking about, right? Or rather, we can put it as B. The first one, what we saw was A was called as the stabilizing selection, right? So B, what we'll be taking up, right? So B is called as directional selection. It is called directional selection. So what happens in this sort of a selection? First thing what you need to remember, as I've already told you in the previous session also, you need to know when each of these selections operate, right? So in which sort of the environmental conditions do these selections operate? The first one, we said that stabilizing selection would always operate in stable environmental condition. Now, where does this sort of a selection, that is directional selection, operate? So directional selection will be operated in the environment which undergoes gradual changes, right? So the environment which undergoes changes gradually, gradual changes in the environment, right? So this sort of a selection usually occurs in the environment which undergoes gradual changes, right? So this is very important, what you need to remember, right? Stabilizing selection would usually occur in a stable environmental conditions where there is no changes happening, right? So in a particular environmental conditions where there is no changes happening, in that sort of environmental conditions, we would have the stabilizing selection. But in a particular environmental conditions where there is gradual changes, slowly some sort of changes are seen in the environment, right? So in those kind of environmental conditions, we have this type of selection being operated, which is called as the directional selection. Now, what happens in this sort of a selection? What happens in this sort of a directional selection? Now, listen it carefully, then we'll get into the picture part, what exactly is happening here. In this sort of a selection, what's happening is individuals present towards one end of the phenotypic distribution are continuously eliminated, right? Let me repeat it once again. So what happens in this sort of a selection is individuals present towards one end of the phenotypic distribution curve are continuously being eliminated from the population. As a result, what would happen now? The mean value, that is the average phenotypic value, will constantly or continuously be shifted towards the other end of the phenotypic distribution, right? So just make a note of it, then we'll get into the explanation part. What does it mean? So what happens in this type of selection is individuals present towards one extreme or one end of the phenotypic distribution curve are continuously eliminated. They are continuously eliminated from the population. So due to this continuous elimination from the population, what is happening is the average phenotype is continuously shifted towards the other end of the phenotypic distribution curve. This is what is happening, right? So what is happening here is, in this type of selection, individuals present towards one end of the phenotypic distribution, they are continuously eliminated, right? So towards one end of the phenotypic distribution, they are continuously eliminated. As a result of which, what is happening is the average phenotype of that particular population is shifted towards the other end, right? So let us see what is happening here. So this is the phenotypic distribution, what we have already seen even in the previous session also, right? So this is the phenotypic distribution curve, what we have, right? And I told you that these are the individuals present in this particular place. They are the ones possessing the average phenotype, right? Now what we are trying to say is individuals present towards one end of the phenotypic distribution, that is individuals present towards this particular area, right? So individuals present towards this particular area are being eliminated. So once the individuals present in this area is eliminated, what is happening to this peak value is, the peak value now, it would actually begin from this place, right? It would actually begin from here and it would come here like this. So this is what is happening here, right? So getting it? Initially, the peak was here, but now the peak has been shifted to this particular place. So why is the peak shifting from this place to this place? Because individuals present towards one end of the phenotypic distribution curve are constantly eliminated, as a result of which what is happening here is the peak is being moving in this particular direction, right? So that's what we have said here, right? So one end of the population, right? So individuals present towards one end of the phenotypic distribution are constantly eliminated. So individuals present here, they are actually said to be constantly eliminated. As a result of which, what is happening? The average phenotype, that is this one, which was present here, has been shifted towards the other end of the phenotypic distribution curve, right? So this is what happens in directional selection, right? So when does this directional selection operate? So directional selection usually operates in an environment which usually undergoes gradual changes, right? So in an environment where there is gradual changes happening, in that sort of environmental conditions, we have this sort of a selection called directional selection being operated. So what's happening in this directional selection? Individuals present towards one end of the phenotypic distribution are constantly being eliminated from the population, such that the average phenotype is continuously shifted towards the other end of the phenotypic distribution curve, right? So this is what we call it as directional selection. So we can take a couple of examples also to understand this sort of directional selection. Well, the best example what we can give here is the example of giraffe, right? So the example of giraffe, right? So when we talk about giraffe, we have already seen how giraffe has been evolved as per Lamarck's theory. What we saw, because of the usage of its some of its organs, such as the neck as well as the forelimbs, right? It got enlarged, as a result of which a longer neck was seen, and that individual or that particular organism got evolved into giraffe. So talking about giraffe, what happens is the average fitness value with regards to the length of the neck, right? Length of the neck was here, that was normal. It got shifted towards a longer neck, right? So the best example what we can give for directional selection is this case of, it is the case of giraffe, where the average neck length shifted towards a longer neck in case of giraffe. One more example what we can take for directional selection is, this also we have studied, DDT resistant mosquito. DDT resistant mosquito. This also we saw when we were talking about the topic called as natural selection. I gave you an example there also, right? So these two can be considered as an example for the directional selection, right? So clear this. Now getting back to differences between stabilizing selection as well as directional selection. First one, what did we say? Stabilizing selection usually operates in a stable environmental conditions, whereas directional selection, when does it operate? It operates in an environment which undergoes gradual changes in the environment. The first difference. Second difference is, in stable selection, what is happening? Organisms or individuals present in this particular peak value, that is, individuals possessing average phenotype, they alone are being selected, whereas organisms this side and this side are eliminated by the nature. Whereas in directional selection, what is happening is individuals present towards one end of the phenotypic distribution are constantly being eliminated from the population. As a result of which, what is happening? The average phenotype is continuously shifted towards the other end, right? So from here, the average phenotype has been shifted to this particular point, right? So there is continuous shifting of the average phenotype towards the other end of the phenotypic distribution curve, right? So the examples what we can give is, we can take the example of giraffe, where the average neck length got shifted to DDT resistant mosquito. right? So this is what we have with the second type of selection, which is called as the directional selection, right? Now let's take up with the second type of selection, or rather the third type of selection. The third type of selection what we'll be taking up is called as the disruptive selection, or it is also called as the centrifugal selection, right? So the third type of selection what we have is called as the disruptive selection or the centrifugal selection. So number C would be termed as disruptive selection, or it is also called as the centrifugal selection. Now what would happen in this sort of a selection, right? So what would happen in this sort of a disruptive selection? First one, as we have been seeing in the last two types of selection, we first started off with in which type of environmental conditions that a particular type of selection operates. Stabilizing selection operates in an environment where there is stable conditions. Directional selection, it operates in an environment which gradually undergoes changes. Here what we are talking about is this type of selection usually operates in an environment when a homogeneous environment changes into heterogeneous type. Right? So this type of selection usually operates when a homogeneous environment changes into heterogeneous type. Now, what do we mean by homogeneous environment and heterogeneous environment? Environment where there is no changes at all, we start calling it as homogeneous environment. Heterogeneous environment where there is different types of changes, or rather we can put it saying that there are different types of environmental conditions in a particular place, that we start calling it as heterogeneous type. So whenever a homogeneous environment changes into a heterogeneous type of environment, in those conditions we have this disruptive selection happen, right? Majority of the cases this type of conversion is actually very rare, so that is the reason why we consider disruptive selection as the rarest of the three types of selections what we have. But even though it is a rare type of selection, it is usually involved in formation of two or more than two different types of species. We'll see how exactly it is happening, right? So first thing what you need to remember is disruptive selection, it operates in an environment whenever a homogeneous environment changes into a heterogeneous environment, right? So whenever a homogeneous environment changes into a heterogeneous environment, we have this type of selection operating. Again, stabilizing selection, when does it operate? Whenever there is a stable environmental conditions, we have the stabilizing selection operating. Second one, directional selection, when does that operate? Whenever there is changes in the environment slowly and gradually, we have directional selection operating. Disruptive selection, when would this be operated? Whenever a homogeneous environment changes into a heterogeneous type, we have this disruptive selection happen. Now, let us see what happens in this sort of a selection, right? So in this sort of a selection, what's happening is individuals present towards both the extremes of the phenotypic distribution are being selected, while the individuals possessing the mean phenotype are being eliminated from the population, right? So just write down, then we'll get into the structural part, right? So what's happening is this is individuals towards both the extremes, right? Individuals towards both the extremes are selected or preserved, while individual possessing average phenotype, individuals possessing average phenotype are being eliminated from the population, right? So what we are seeing here is individuals present towards both the extremes, they are selected, right? They are selected, while the individuals possessing average phenotype, they are being eliminated. So if you just observe this, it is almost opposite to that of the stabilizing selection what we have seen. So what did we see in stabilizing selection? individuals towards both the extremes are eliminated, while individuals possessing average phenotype are being selected in case of stabilizing selection. But in case of disruptive selection, what is happening is it is quite reverse, right? Where individuals towards both the extremes are selected, while the average phenotype are being eliminated from the population. Again, let's take up the phenotypic distribution curve, right? So this is the phenotypic distribution curve what we have, right? And I told you that this would be the peak value here. Individuals possessing the average phenotype is actually present here at the center. Now what is happening? Individuals towards both the extreme, they are selected. Individuals present here and here, they are selected, while individuals possessing average phenotype, that is this individuals here, they are actually eliminated from the population. Now once these individuals are eliminated, what would happen is, right, it would lead to the formation of two or more different types of subpopulation, right? So this would now become somewhat like this, right? The graph what we saw now, it would become somewhat like this, right? So this is one extreme end and this is one extreme end. Individuals present towards the center, that is possessing the average phenotype, are being eliminated from the population. Now, as I've already said, this type of selection is actually considered to be the rarest of all the three different types of selections what we have. But they have the ability to lead to the formation of two or more subspecies or two or more subpopulation, right? So this type of selection, right? So this type of selection would lead to the formation of two or more subspecies or two or more subpopulation, right? So this is what is seen in case of disruptive selection, right? Now here we have one subpopulation and here we have one subpopulation. Now, if these two subpopulations do not interbreed among themselves, right? Try to understand this. If these two subspecies here and here, if they are not able to interbreed among themselves, we call it as reproductively isolated individuals. Now, these two would become reproductively isolated, as a result of which this would become one population, one species, and this would become another species, right? So that's how they say different species have been evolved, right? So quickly going through this disruptive selection once again. So what is this disruptive selection all about? So when does it operate? So this disruptive selection usually operates in environmental conditions whenever a homogeneous environment changes into a heterogeneous type. Secondly, what exactly happens in this sort of a selection? Individuals present towards both the extremes of the phenotypic distribution are being selected, are being preserved, while the individuals possessing the average phenotype are being eliminated from the population. As a result of which, what is happening? This would lead to the formation of two or more subspecies or two or more subpopulation. Now, if these two or more subpopulation do not interbreed among themselves, we call them as reproductively isolated individuals, and these reproductively isolated individuals would now lead to the formation of a new species, evolution of two or more different types of species. This is what we have with disruptive selection. So what examples can we give here for disruptive selections, right? One best example what we can give for disruptive selection here is the example what we took up when we were studying about industrial melanism, right? So the examples here would be the black and white peppered moth, right? So the black and white peppered moth were actually formed through this sort of disruptive selection, which we call it as Biston Betullaria, right? So Biston Betullaria has been selected, and that sort of a selection is based on disruptive selection. One more example what we can give for this here is Darwin's finches. Darwin's finches, right? So we saw when we were talking about the theories, right? We saw that almost one particular species of bird, right, which was seed eating, got evolved into 13 different types of species in the Galapagos Island. And that 13 different species origination or evolution is again through this sort of a selection called the disruptive selection. One more example also is given in our books, right? The sunflower in case of California, right? So the sunflower in California, through this sort of a selection called as disruptive selection, it got evolved into two species, where one species were able to grow in a dry land, whereas the other species were able to grow in a wet land, right? So this is what we have with the disruptive selection, right? So three different types of selections what we have seen so far pertaining to the process of natural selection. The first one is stabilizing selection, right? So what happens in stabilizing selection? Individuals present towards both the extremes are being eliminated from the population, whereas individuals present towards the mean phenotypic value, they alone are being selected. That is stabilizing selection. Second type of selection what we saw is the directional selection. So what happens in directional selection? Individuals present towards one end of the phenotypic distribution are eliminated, such that the average phenotype constantly shifts towards the other end of the phenotypic distribution. Second type of selection, which was directional selection. The third type of selection, disruptive selection. So when does this occur? Whenever the environment changes from homogeneous type to a heterogeneous type, we have this sort of a selection. And what happens in this sort of a selection? Individuals present towards both the extreme here and here, they alone are being selected or preserved, while the individuals possessing the average phenotype are being eliminated from the population, right? So this sort of a selection is called as the disruptive selection. So this is the first evolutionary force what we have, which is called as the natural selection, which brings about some sort of changes in the gene pool of a particular population, right? I hope this part is clear. The first type of selection, the first type of evolutionary force, which is called as the natural selection, right? Now, let's get into the second type of evolutionary force, right? So the second type of evolutionary force, what we'll be taking up, right? So the second type of evolutionary force, what we'll be taking up, is called as gene flow, or it is also called as gene migration, right? So let us take up with the second type of evolutionary force. The second type of evolutionary force, first one was natural selection. The second one is gene flow, which is also called as gene migration. I hope all are familiar with the terms flow as well as migration. So what does this mean, migration or flow? It is nothing but movement. So what happens in this sort of a evolutionary force is, whenever the alleles, the term gene flow could be defined as the movement of alleles from one population to another population, right? So what is this gene flow? It is nothing but the movement of alleles from one population to another population, right? The movement of alleles from one population to another population is actually called as the gene flow, right? So what we are dealing here is, we are talking about the changes in the gene pool. The second factor which affects or brings about the changes in the gene pool is this gene flow. So what's happening in this gene flow? There is movement of alleles taking place from one population to the other population. Now let's just imagine that this is one population, population A, and here there is another habitat with population B. Here there are some individuals, right? So say 1, 2, 3, 4, 5, 6, 7, and 8, right? Here there are some individuals A, B, C, D, E, F, G, and H. These are all the individuals present in both the population. Now, say one of the individuals, say individual number 3 or allele number 3 is moving out of the population. It is moving out of the population. Now, this movement of allele 3 out of the population, right, is called as migration. More precisely speaking, we start calling this sort of a migration as emigration, right? We usually call it as emigration. I hope all are familiar with these terms. Emigration. Now, due to this emigration, what is happening? The gene pool starts changing. So, what do we call it as? What do we mean by a gene pool? The sum total of all the genes present in a sexually reproducing population. We started calling it as gene pool. right? So all the genes, or rather all the alleles present in this particular population would give us something called as a gene pool. Now, when one of the gene is moving out of the population, what would happen? Definitely, the gene pool will start decreasing. The number of genes and the number of alleles here will start decreasing, thereby bringing about some sort of change in the gene pool of this particular population. So this is through the process of immigration. Now, when the same allele moves into population B, right? So the allele has moved here into population B. Now, this movement of allele from here into population B, we start calling it as immigration, right? So we start calling it as immigration. Now, what's happening through immigration? There is one gene added to the population B. So in the first case, what is happening? One gene is being removed from the population. In the second case, one gene is being added through the population. So through the process of emigration, the genes are being removed from the population, right? Or there is loss of genes from the population. Whereas through the process of immigration, the genes are being added to another population. This again brings about changes in the gene frequency in both population A as well as population B. So this is what we call it as gene flow or gene migration. Right? So what is this gene flow or gene migration? It is nothing but the movement of alleles from one population to another population is called as gene flow, or it is also called as gene migration. So we also use the term as this population here would be termed as donor population because it is donating one of its gene, and this population here would be termed as a recipient population because it is accepting one of the genes. So these two conditions, donor population as well as recipient population, what is happening? There is some sort of change in the gene frequency as well as the gene pool also. Right? So this is the second factor what we have, which is called as gene flow or gene migration. Right? Now, moving on to the third factor what we have. Right? Moving on to the third factor, which is called as genetic load. So we'll see what is this genetic load all about. So the third thing what we'll be talking about is called as genetic load. So number 3, the third factor is called as genetic load. So first take down the definition of genetic load, then I'll tell you what exactly is genetic load all about. Right? So genetic load could be defined as the existence of a deleterious gene, in a population is called as genetic load. Right? So what does genetic load mean? Existence of a deleterious gene in a population. Now what do we mean by this term? Deleterious gene. Deleterious gene could also be given as a harmful gene or a disadvantageous gene. Right? Either a harmful gene or a disadvantageous gene is usually referred to as a deleterious gene. Now, if that sort of a harmful gene or a disadvantageous gene is present within a population, that condition we usually call it as genetic load. Right? So what is the genetic load? Existence of a deleterious gene in a particular population is called as a genetic load. So we'll take one simple example. So once we study this example, I think you'll be very clear with what is this genetic load and what is the deleterious gene all about. The famous example, what we have already studied several times, which is called as sickle cell anemia. Right? Sickle cell anemia. So we have already discussed about this disorder, sickle cell anemia, right? A genetic disorder which is caused due to mutations in the gene, right? Which codes for beta polypeptide chain of the hemoglobin molecule, right? So what happens in this sickle cell anemia, right? So what happens in this sickle cell anemia is the individuals who are homozygous for sickle cell, right? So the individuals who are homozygous for sickle cell. So how do we denote it? HBS and HBS. So these are the individuals who are homozygous for the sickle cell gene. Now these individuals, as we have already seen, what would happen? they will not survive. They usually die during their early childhood days itself because of this particular mutation and presence of the sickle cell RBCs. They will usually die during the early childhood days through because of a condition called as anemia. Whereas when we see the heterozygous condition, right? When we see the heterozygous condition of this particular gene, how do we denote it? HBA and HBS, right? So this is the heterozygous condition of the sickle cell gene. Now, this individuals possessing this sort of a gene also, we know that they usually live reasonably healthy, provided they have enough of oxygen. That's what we have seen when we were discussing it in the topic called as genetics. All right. Whenever these sort of individuals, they start facing low oxygen tension, at that point of time, this allele also would be mutated, again leading to sickle cell anemia, and they usually die. But if they have enough of oxygen, even though they are possessing this sort of a heterozygous condition, they will live reasonably healthy. But even though they live reasonably healthy, they have to maintain proper conditions. That is the reason why this sort of a gene, the heterozygous condition of the sickle cell anemia, would be regarded as a deleterious gene or a harmful gene, right? Or it is also considered as a disadvantageous gene. Why it is a disadvantageous gene? Because anytime whenever this individual faces low oxygen tension, at that point of time, there is a chance that this particular individual also would start producing excess of sickle cell RBC, as a result of which they also would die. So the presence of a deleterious gene in a population is what we call it as genetic load, right? So the presence of a deleterious gene in a population is called as genetic load. So this is the third factor what we have, right? Third factor what we have with regards to the evolutionary forces. The first one is natural selection. Three types of natural selections we saw. One is stabilizing selection, second one is directional selection, and the third one is disruptive selection. The second thing what we saw is, right, the second one what we saw is the genetic flow, or rather gene flow or gene migration. So this also would start changing the gene frequency or the gene pool. It would bring about some sort of changes in the gene pool. And the third one, what we are seeing here, is the genetic load. The third factor associated with the environmental forces, what we have. Right. So this is what we have with genetic load. Now, moving on to the next one, number 4, would be called as genetic drift. So we'll see what is this genetic drift all about. Another important factor here, right? Factor number 4, which is called as genetic drift. Now, what is this genetic drift all about? Right? So, what is this genetic drift all about? So, first take down the definitions, then we'll get into the explanation part. Right? So, the changes in the gene frequency, right? So, we can put it this way: random changes. A few important things what you need to remember here: random changes in gene frequency. Random changes in gene frequency in small population caused due to chance events and not by natural selection. This is actually called as genetic drift. Right, so important one, what you need to remember here, genetic drift. So what is a genetic drift? The random changes that occur in the gene pool of a small population due to chance events and not by natural selection. This is called as genetic drift. So three important things what you need to remember whenever you talk about genetic drift. Right? So three important things. What are the three important things? First one, random changes in the gene frequency. The first one, random changes in the gene frequency. So where is this random changes in the gene frequency happening? Another important one, always in a small population. So whenever you find the term small population, automatically you can link it up with genetic drift, because genetic drift would be seen only in case of small population. right? So the second one is small population. First one, random changes in the gene frequency. Second one, in a small population. The third important thing is which is caused by chance. It is caused by chance and not by natural selection. Right? So the changes in the gene frequency, which is because of some sort of the chances, but not by natural selection. And that too in a small population is called as genetic drift. So how do we define genetic drift again? Any sort of random changes that occurs in the gene frequency in a small population, and that too it is by chance and not by natural selection. So this is called as the genetic drift. So let us take a simple example to understand this genetic drift. Right. Let us talk about a small population. Right. So let us consider a population where there is one particular allele having, one particular gene having two alleles. So we have a population, right? In a population, one gene, and that gene, say it has two alleles. Now, the allele frequency of one type of allele, right? So we are talking about the allele frequency. So the allele frequency of one type of allele is say 1%, right? So the allele frequency in a for one particular type of allele is only 1%. Now let us consider two populations. One is a large population and a small population. The large population, say there is 1 million alleles. There are 1 million alleles in a large population. Now when we say 1 million alleles, 1% of 1 million would be how much? It would be 10,000 alleles. Right. Let us take a small population now. Say the small population has 100 alleles. So 1% of 100 would be 1. Getting it? Right? So we are trying to explain this genetic drift, concept of genetic drift. So what did we say is a genetic drift? It is a random changes that occur in the gene frequency in small population. Why we are taking small population? Through this you'll get a clear picture. Right? In a small population, due to chance event and not by natural selection is called as genetic drift. Right. Now, let us assume that in a particular population, there is one particular gene having two alleles. Let's consider that the allele frequency of one particular type of allele is 1%. So we are comparing two different populations now. One is a large population having 1 million alleles. So 1% of 1 million would be 10,000 alleles. Right. Next, small population having only 100 alleles. 1% of 100 alleles would be only 1. Now, the probability, right, try to understand this now. The probability of losing the allele will be more in which of the two populations now? Getting it? Right? So, the probability of losing the allele would be more in which of these two populations? Obviously, it would be for this sort of a population, right? So the probability of losing the allele would be more in a small population rather than a large population. Getting it? Why we are saying that the probability of losing the allele is more here in this small population than the large population is. We know that losing one allele out of 10,000 will not make much difference, right? Getting it? Losing one allele out of 10,000 alleles will not make much difference. But losing one allele out of one will make a huge difference in the population. Get it? Losing one allele out of 10,000, right? Losing one allele out of 10,000 allele will not make a huge difference. Will not make much difference at all, right? Because there are almost more than 9,999 already are present there. But losing one allele from only one will make a huge difference. So that's what we mean by genetic drift, right? So random changes in the gene frequency in a small population due to chance events and not by natural selection, right? Why? Because the probability of losing an allele, right, losing one allele is more in case of small population than the larger population, right? Now what would be the end result of this? The end result of this assumption what we have taken is would be either something called as fixation, right? It would be either fixation, where in fixation P or Q, that is the dominant allele frequency or the recessive allele frequency would be 1 because of fixation, what we call it as addition, right? And in the second case where it could also lead to loss of the allele where P or Q would be equal to 0, right? So we start calling it as either fixation where P or Q would be equal to 1. Second thing, we can also call it as loss where P or Q would be equal to 0. So the end result of this assumption, what we have taken is, it could be either fixation or it could be loss. If it is fixation, it would be P or Q would always be 1. If it is loss, P or Q would be 0. Right? So this is what we have with the topic called as genetic drift. So what is this genetic drift all about? The random changes that occur in the gene frequency of a small population due to chance events and not by natural selection is called as genetic drift. So a simple example we have taken up to understand what is this genetic drift and why it occurs only in a small population. Right. Now, we have two different types of examples under genetic drift so that we can understand more about genetic drift. Right. So there are two important things or two important examples what we have under genetic drift. The first example what we'll be studying is called as the founder's effect. Right. So this is the first example what we'll be studying. It is called the founder's effect. And the second example what we'll be studying is called the bottleneck effect. Right. So we have the founder's effect as well as the bottleneck effect. So these are the two important examples what we'll be studying under genetic drift. Right. So the founder's effect as well as the bottleneck effect. So four characters or four evolutionary forces we have seen so far. One is natural selection. Under natural selection, we spoke about the stabilizing selection, directional selection, and the disruptive selection. The second one, what we saw was genetic load. The second one, what we saw was gene flow or gene migration. The third one, what we saw was genetic load, and the fourth one, what we are taking up, is called as genetic drift. Under genetic drift, we have two more here. Two examples here. One is called founder's effect and the other one is called the bottleneck effect. Right? So we'll stop here for today. In the next session, we'll take up in detail what is this founder's effect and what is this bottleneck effect. And along with that, two more, just a brief introduction to that because we have already studied several times about it, mutation and genetic recombination. So with that, we'll be done with the topic called mechanism of evolution. Right? So that remaining part of the mechanism of evolution as well as the evolutionary forces, we'll take it up in the next session. Fine? Thank you.